Imagine you're a soldier on a critical mission. Your objective: take out the enemy combatants who are hiding deep inside a crowded city. Traditional chemotherapy is the grenade. You throw it into the city and it blows up everything in its path. It is effective. It does indeed kill the enemy, which in this case are rapidly dividing cancer cells. But here's the problem. The explosion is not targeted. It also destroys everything else that happens to be moving fast. Your hair follicles, your stomach lining, your immune cells. This is why chemotherapy makes patients so sick. It is collateral damage on a massive scale. Now, imagine you have a sniper rifle. You can identify exactly which building the enemy is hiding in. You take one shot, one target, minimal collateral damage. That is exactly what antibody-drug conjugates, or ADCs, are designed to do. They are chemotherapy with a GPS tracking system, a sniper rifle in a world of grenades. In this video, I will break down how these molecular snipers work. We will explore the biology behind the targeting system, the chemistry of the payload delivery, and most importantly, why the hardest part of this technology isn't the biology, but the chemistry. And towards the end, I will share something that genuinely surprised me when I started reading the priming literature. The assumptions that we have made about how these drugs should be designed might be completely wrong. Let's start with the first piece of the puzzle: how the ADC actually finds the cancer cells. Cancer cells are fundamentally different from healthy cells. They display specific proteins on their surface, proteins that healthy cells either do not have at all or have in much smaller quantities. These proteins are called antigens. Think of them as flags, giant waving flags that say, "Attack here, I'm different." Scientists have learned to exploit these flags. They engineer monoclonal antibodies, Y-shaped proteins that act as biological keys. These antibodies are designed to lock onto one specific antigen found on cancer cells, and only that antigen. So, the antibody is our GPS. It guides the entire ADC directly to the cancer cell and nowhere else. This can prevent the drug from killing other fast-growing, healthy cells. So, we have our GPS, the antibody, and we have our weapon, a highly potent chemotherapy drug. But how do we attach them together without breaking either one? This is where the linker comes in. The linker is the chemical bond that connects the antibody to the drug. Think of it as a safety pin. Scientists attach a highly potent chemo drug to the antibody using this linker. These drugs are so toxic that they cannot be administered on their own. They would kill the patient before killing the cancer. But when attached to the antibody via the linker, they are safely transported directly to the cancer tumor. However, this linker must be completely stable in the bloodstream on the way to the cancer. If the drug falls off early while the ADC is still circulating, it becomes toxic poison floating freely through the body. This causes side effects, and this phenomenon is called off-target toxicity. The moment the ADC enters a cancer cell, specifically the cell's lysosome, which is highly acidic with a pH of around 4.5 to 5, the linker must instantly break open, releasing the drug to kill the cell from the inside. Researchers have designed several different types of these linkers that attach the antibody to the drug, each with its own mechanism of release. Here are some examples. Acid label linkers are designed to break in low pH environments. They are stable at pH 7.4 in the blood, but hydrolyze rapidly at pH 4.5 in the lysosome of the cancer cell. Enzyme cleavable linkers are chopped by specific enzymes that are highly active inside cancer cells. Redox-sensitive linkers break when they encounter high levels of glutathione, a molecule that is much more abundant inside cancer cells than outside due to their increased metabolic activity. For decades, the dominant assumption in ADC research was simple and intuitive. If we just make the linker more stable, we will have a better drug. The logic seems obvious. A more stable linker means less premature drug release in the bloodstream, which means fewer side effects. So researchers poured billions of dollars into developing ultra-stable linkers. It seemed like a straightforward engineering problem. But here is when I started questioning this assumption after reading a 2026 paper published in the Annals of Oncology. The paper re-evaluated the clinical data of all FDA-approved ADCs. The researchers looked at the linkers used in each drug, their stability profiles, and most importantly, their clinical outcomes. And the findings genuinely surprised me. Some of the most successful FDA-approved ADCs actually have relatively unstable linkers, yet it works exceptionally well clinically. It has remarkable efficiency and a manageable safety profile. On the other hand, ADCs with ultra-stable linkers, the kind that were designed to minimize premature release, have not consistently shown better outcomes. In fact, some of these super stable ADCs have been associated with unexpected toxicities like severe skin reactions, lung inflammation, or even new types of side effects that were not predicted from the preclinical studies. The paper also pointed out a major limitation in ADC development. Preclinical models do not predict human outcomes well. A linker that looks perfectly stable in mice might shed its payload rapidly in humans due to differences in metabolism. Mice and humans process these drugs differently, and the gap is often large. This made me realize something important. Maybe the problem is not simply linker stability. Maybe the relationship between linker stability and clinical success is more complex than anyone assumed. So, what if an unstable linker is actually better? When I started researching ADCs, I assumed it was a straightforward engineering problem. Design a more stable linker, get a better drug. It seems so logical. But reading this paper flipped my perspective. I learned that reality is sometimes messier than theory. My question is now this. Are we overcomplicating linker design? Are we pouring billions of dollars into making linkers more stable when the real bottleneck might be something else entirely, like how the drug is metabolized, how the ADC distributes in the body, or how the tumor microenvironment affects drug release? I don't have the answer yet, and honestly, I don't think anyone does. If you have read papers on this topic or have related thoughts, please feel free to let me know in the comments below.